Let's understand the physiology of Loop of Henle. There are two parts of Loop of Henle. The first one is descending limb going deep down in medulla region and ascending limb going up toward cortex region. Descending limb is only permeable to water, contains aquaporins, while ascending limb is permeable to solute, contains sodium-potassium 2 chloride cotransporter. Osmolality of filtrate in proximal tubule is same as blood, which is 300 milliosmoles, that is isotonic with blood. It's because 65% of both sodium and water is absorbed in proximal tubule back to blood. As the filtrate go down through Loop of Henle in medulla region, its osmolality starts increasing from 300 milliosmoles to 1200 milliosmoles. It means filtrate here is very salty and hypertonic as compared to blood. Let's see how filtrate get hypertonic here. Filtrate coming from proximal tubule contains sodium-potassium, chloride, magnesium, calcium and water. At first, in ascending limb, sodium-potassium chloride cotransporter pumps sodium, potassium and chloride from the filtrate into the tubular cells. After entering inside cell, sodium will be pumped into the interstitial space through sodium, potassium ATPase pump. Chloride will also move into the interstitial space through chloride channels at the basolateral membrane. Potassium that enter into cell will leak back into the tubular lumen, making the lumen positively charged. Calcium and magnesium present in the filtrate will repel by the positive charges, move through the paracellular space between the cells into the interstitial space. Urea is also transported to this interstitial space from collecting duct. Now you can see that sodium chloride, magnesium, calcium, urea all are present in interstitial space, making it very salty. You know water always follow to more salty area, so water in descending limb will move from aquaporins into this salty interstitial space. Water keep moving until osmolality balances. This is called countercurrent multiplier mechanism, because filtrate is flowing in opposite directions in the loop. As water is leaving from the descending limb, filtrate here becomes more hypertonic. And as solute is being absorbed from the ascending limb, filtrate here becomes more hypotonic and diluted. This explains why osmolality of filtrate is around 200 milliosmoles as it leaves the ascending limb and reach the distal tubule. About 25% sodium and 15% water is absorbed here. In this region blood is provided by Vasa recta, branch of efferent arterioles. Blood flows very slowly through Vasa recta in opposite direction. As blood goes down through the descending limb of Vasa recta, it picks solute like sodium chloride and drops water into this salty space. And as blood goes up through ascending limb, it drops back sodium chloride and picks back water from this salty space. That's how Vasa recta preserve this medullary interstitial gradient, prevent rapid washout of salts and removal of sodium chloride. This is called counter-current exchange of Vasa recta. Beside this, Vasa recta also supply nutrient and oxygen to this region.